Hello, and my name is Mary Regina Boland. I'm an assistant professor of informatics in the Department of Biostatistics, Epidemiology, and Informatics at the University of Pennsylvania. I'm here to describe my work and the work of my lab on developing methods to study maternal and child health outcomes, including genetics and environmental contributions. Our lab develops methods to utilize various types of data from clinical records. This includes high-dimensional clinical data from both outpatient and inpatient settings, data from clinical text, which we need to develop methods using natural language processing to help address, environmental data, which we need to link using external sources back to the clinical data, often using address information and other geospatial coordinates, as well as genetic data, which is linked to the clinical data, and we use a resource called the Penn Medicine Biobank in these research. Our ultimate goal is to study human health and disease and learn novel clinical, genetic, or environmental links that shed light on diseases and their etiologies. This is an example of what the electronic health record data looks like when clinicians are entering it. They can enter it via iPad or directly to the clinical record on a computer using a kiosk system. Everything is recorded in electronic health records. These data are recorded using International Classification of Disease codes. Here we show a few examples of ICD-10 codes, and personally I find it incredible how much information and detail is recorded in electronic health records. For example, if you're hurt at the library, there is a diagnosis code for that. If you walk into a lamppost, subsequent encounter, that is also a diagnosis code, as well as things like being struck by a macaw or a sea lion. How much data do we have? So at Penn Medicine, we are actually very fortunate to have data from 6 distinct hospitals in the Philadelphia metropolitan area. 3 are directly in Philadelphia itself, 1 is in Lancaster, another is in Princeton, New Jersey, and a 6th one is also located in Chester County, which is located very close to Philadelphia. We are able to use the data in Philadelphia from the 3 distinct hospitals to actually train informatics models using data from 2 hospitals within West Philadelphia and then validating on a 3rd hospital located in inner city Philadelphia. So we have a very rich source of data from multiple EHR systems in the Philadelphia metropolitan area. Data from Penn Medicine actually comes from over 1,048 different clinics. This includes data from both inpatient and outpatient settings. These data are very diverse. Here we have actually within each N equal sign, we have the number of distinct clinics belonging to that category. So for example, there are 89 distinct OBGYN clinics within Penn Medicine, and there's 104 distinct cancer clinics. Many of these different types of cancer clinics actually treat different types of cancer, and therefore they are very different from one another. However, the category in general of cancer clinics is similar and yet also distinct from OBGYN clinics. When using electronic health record data, we often need to be able to distinguish where the information is being recorded to understand the quality of information that is being derived. So for example, if information on your pregnancy is coming from the OBGYN clinic, we can trust that data a little bit more than, for example, information that may be coming from a cancer clinic about your pregnancy. In this way, we can synthesize data in electronic health records from across the entire Penn Medicine system as a whole in order to learn the appropriate clinical information for each patient, and this also then has to be transformed into our models so that we understand the correct clinical history for each distinct patient. In our lab, we also develop methods to analyze clinical text because often these disease category codes like ICD-10 that I previously mentioned, they do not capture the rich enough information that we need. One important problem when understanding clinical notes is acronym disambiguation. Acronyms are used in clinical notes all of the time because these help doctors write very short notes, which is very important given the amount of time that they actually have to write the note. On the right-hand side here, we have the number of definitions per acronym from a tool that we developed called Classy Gator, which stands for the Clinical Acronym Sense Disambiguator. This work was part of Aditya Kashyap's doctoral research. He's a current PhD student at computer science, jointly mentored by both myself and Dr. Chris Callison-Birch. And his method helps to disambiguate the meaning behind each acronym usage within clinical notes. And this helps us to begin to understand some of the clinical information that's packed into these clinical notes. Our lab also develops methods to integrate and understand environmental exposures. In order to do this, we must combine data from multiple data sources, many coming from the U.S. government. These include the USGS, which is the United States Geological Survey, the FDA, which is the Food and Drug Administration, EPA, or Environmental Protection Agency, the CDC, Centers for Disease Prevention and Control, as well as the USDA, NOAA, and NASA, and they're all listed there. The data I previously mentioned on environmental data is very, very helpful in understanding the relationship between birth season and disease risk. This image here shows the location of sites that have been used in a few different research papers from my lab, investigating the role of birth season and disease risk from across the globe, including 2 sites in Asia. This work allowed us to reveal that the first trimester exposure to air pollution increased the risk of atrial fibrillation in 10.5 million humans. Upon coming to Penn Medicine, we were able to expand this research and look at the role of air pollution during development on the risk of cardiovascular disease in 130,000 dogs. That's shown on the right. Overall, validating these different associations between the humans and dogs further increased our ability to have confidence in our findings, and also strengthens the fact that we are able to develop methods to help individuals to understand many complicated things, including their personal exposure to hormonal modulating chemicals, which is the next focus of our research. We've looked at hydraulic fracturing fluid and its role on impacting various hormonal pathways. For example, the map shown here indicates states in the country where there's a high proportion of estrogen wells. The state of Alabama has a large number of estrogen wells, or a large proportion of estrogen wells that use hydraulic fracturing fluid that target estrogenic compounds. Hydraulic fracturing wells that target testosterone pathways are shown here in bright yellow and include Ohio, Pennsylvania, and Illinois. This work has actually been developed into an app, and I will briefly show you what it looks like here in this little movie. You can type your zip code information into the app and then locate wells near you that target a particular, either have a particular name or affect a specific gene or ingredient that you're interested in. For example, if you're interested in estrogen, general hormones, or testosterone, you're able to click them as shown here. You can also include only toxic chemicals, and those are identified using the Agency for Toxicity. And then you can click on each individual well as well, and it will provide you with the list of ingredients as well as further information on each of the ingredients and whether or not it is a food compound regulated by the FDA or not. Our lab also does work on maternal stress, disparities, and drug response, and one of the principal members looking at this is Jessica Meeker. She's a doctoral student in epidemiology in my lab, and she's developing methods which can be used by others within the lab to identify patients who move and therefore change geolocation during pregnancy, as well as to assess their level of stress at both the individual and community levels, and the relationships between stress and drug response. Dr. Sylvia Canelon is a postdoctoral scientist in the lab, and she's written a in-depth and very detailed literature review on climate change and its role in women's health. This includes how various weather effects can affect food availability, as well as toxins released in the environment, and that all of these factors taken together can perturb timing in menarche. Altered timing in menarche can increase the risk of 4 different disease categories, including mental health issues, fertility-related problems, cardiovascular disease, as well as bone health. In addition, Dr. Canelon has worked on developing MADI, which is the method to acquire delivery date information from electronic health records. And this is an algorithm used by our lab to identify a cohort of pregnant women who have delivered at Penn Medicine. We were able to validate this algorithm against a gold standard birth log, and we are very confident in these results. And this cohort of women can be used for further research. Pee Wee was a summer intern in our lab through the Leonard Davis Institute's summer internship program last summer, and she was able to correlate preterm birth as an outcome, which was identified via Dr. Canelon's algorithm, and housing quality within the city of Philadelphia. We hope to further expand upon our methods to develop methods to link Penn Medicine Biobank genetic data to help us understand maternal-fetal outcomes. I show here a screenshot from Ancestry DNA showing a person's result regarding birth weight. So there are genetic factors that are known to increase your risk of being an average-sized newborn or a very large newborn or a very small newborn. This is just scratching the surface, the tip of the iceberg with regards to the genetic factors and the role that they play in maternal as well as fetal health outcomes. We wish to explore this further using the Penn Medicine Biobank, and we actually need a rotation graduate student to work with Penn Medicine Biobank as well as us to help us uncover patterns correlated with maternal health outcomes. So please let us know if you are interested. In conclusion, our lab works to develop methods to understand the role of environmental exposures on maternal and fetal health outcomes. Specifically, we look at hydraulic fracturing fluid, PFAS, which is a chemical that I have not gone into during this presentation, as well as other climate variables, including temperature, and social variables such as housing and violence in Philadelphia. Our clinical outcomes that we specifically focus on are maternal morbidity and mortality, as well as cardiovascular disease, sickle cell disease, and birth season-dependent diseases that we have identified via prior research. We're hoping to connect genetics with many of these above outcomes, and we're interested in PhD rotation students who are interested in exploring opportunities with us. If you're interested in any of these outcomes plus genetics, please come and talk to us further. This is an overview of the members of our lab, and because of COVID, we haven't been able to meet together, but in 2019, pre-COVID, we had a couple of outings, and hopefully in 2021, we would be able to get together again physically without the need for social distance. Thank you very much, and please reach out to me if you have any questions of any kind.